It's Friday night, and that means we're back again for another week of Bioactive Live Q&A. We're here again with another special guest, Organic Alexa, coming back on for Microscopy Soil Part 2. We're going to kind of slow it down a little bit this week and take it kind of piece by piece. Look at some healthy soil, look at some unhealthy soil, teach you guys what it is that you're looking for in those healthy soils. But first, my name is Mark. I'm your host. And with me, as always, I have my co hosts, Brian from Rubber Ducky Isopods and Kevin from OK Calyx. It is a pleasure to be back with all of you again. Organic Alexa, or Alexa, we'll call you. Um, how are things? Anything new since the last time we spoke?
Oh gosh, yeah. I mean, I'm going through lots of changes right now. So like thank you to everyone who has followed me and sorry for the lack of posting, but I'll be back at it once I settle into these new life changes. But no, yeah, everything's been good and I appreciate the opportunity to come back on. Like you said last time, I kind of was explaining, some really more in-depth things. And yeah, this time I think it will be nice to kind of back up and start from the beginning of the story so we can have context to what we're talking about. And yeah, I have some really nice examples of different samples to go over and share. And yeah, we can just discuss kind of what's happening in there and how, certain samples may be more beneficial than other samples. And yeah, just explain what's going on.
Yeah. Yeah, now make no mistake about it. The last episode that we had with you, I believe it was episode 26. If you haven't watched it, please go back. Make sure you hit the like button. Make sure you hit the subscribe button. This is a small non-profit show that we're just putting together for fun. And we appreciate everybody that follows the people that come on here because we're trying to all boats rise together. So huge ups. Follow Alexa at Organic Alexa. But episode 26, we went through a lot of amazing information and we had a ton of questions. I think it was like one of the longest episodes we've ever had. It was almost 2 hours long and it was great conversation. But like you said, it was kind of all over the place because questions, and we were also trying to do something completely new, which is throw a living sample of Kevin's IMO3 under a scope and have you chase around microorganisms under 200x or whatever it was that we were looking at, which is
Right.
we did that again with Andrew from at Biotactics
Oh, really?
from Benamite. Yes, it wasn't as successful. It's a lot harder to chase around predatory mites, apparently,
Right.
Californicus and stuff. Yeah. So,
Equipment
um,
working on him too, didn't it? Right at the beginning,
say that again, Kev.
didn't his equipment even quit working on him or something like
And in round two, his equipment completely didn't work. So we were completely
We're
kind
ready
of
for a
free.
good show, Alexa. That's what we're saying.
Well, it's
Um,
the sample,
yeah,
honestly. Sometimes you never know what you're gonna get with samples. Sometimes it's hard to do
exactly,
with life
exactly.
microscopy, but when you get a really good sample where it's like every field of view, there's something going on, which is pretty rare, honestly.
Oh,
So
and yeah, and Kevin, you're right. Actually, now that I remember correctly, I had to throw my sample underneath a scope and hit the batch of spider mites I had with me. It went out to a buddy's greenhouse, came back, sat in hot cars, so like half of it was completely toasted, but it was stuff that he had sent me that I was gonna bring back. I just forgot, like I was like a complete idiot. So the sample wasn't even amazing either. Like there was a lot of bodies. It wouldn't be what you get fresh. So I guess the point I'm trying to make is live microscopy is very, very hard. That's why we're going to kind of slow this down, take a step back, and start with healthy soil versus your standard kind of sterilized ag soil, agricultural soil that you would find in the field. Do you want to start with that, Alexa? Maybe start with what we'll find, or let's start with what a dead sample looks like,
Yeah,
I guess.
that's kind of what I was thinking. So I hit present. Is that what I do?
Oh, yes. Hit
Okay.
present and
Oh, share
once
screen. Oh, share screen. Share.
and this is part of being live is I totally forgot about setting this up beforehand.
No worries. Okay. So can you see what's going on?
Yes.
All right. Awesome. Okay. So, this is as large as I can make it. Can you see everything?
Yep.
Yep.
Okay. Perfect. Okay, so here is a conventionally farmed agricultural soil from one of my clients. This is something that, has been treated completely conventionally: pesticides, fungicides, herbicides, fertilizer, conventional fertilizer applications. And honestly, this is like probably not, or this is like not as bad as I've seen them. There is still a little bit of fungi in here, and
Is that what you see with the light silhouettes?
here, here, like this little piece right here, this brown piece. Can you see my like cursor?
Yep.
Yep.
Okay. So this little brown piece here, that's a piece of fungi, but like primarily you're really just seeing bacteria and like mineral particles. Something else to notice is that there's not a whole lot of like organic matter aggregates. So that's these things here. When we flip to the other sample, you'll see a lot more of those. But it's just primarily bacteria, not a whole lot of anything else. And that's kind of what happens, when we treat our soils in this way. It, kills off the microbial biodiversity, and really you're creating conditions where only like bacteria can really survive. So that's kind of what it looks like. And, my whole goal with what I do as a consultant to improve soil health is to, improve the biological diversity in soils so that, we don't need to use the pesticides, the fungicides, and in the conventional fertilizers. So let's see. This is a cedar forest soil. So this is kind of what I model all of my like compost and compost tea recipes after. This is what I aim for because this is, the most active, the most diverse my ecosystem, soil ecosystem that I have looked at under the microscope or found. I'll take samples from just like healthy ecosystems or unhealthy ecosystems and all kinds of various places. And this, I guess I call it like a cedar swamp, but there's also birch and hemlock, and it's very diverse, lots of different plant species and tree species. So I'm like, okay, this is pretty much the healthiest ecosystem that I can find. So this is what I want to like model my soil after. Um, so let's just kind of
So,
start from the
Alexa,
beginning
I
and
interrupt
story.
you.
And
How do you go about modeling this? What are the things you're adding or trying to do to make it similar?
So, you know, first you want to start with, an inoculant that has, these organisms in it. So, there's lots of different ways you can do that. Getting leaf mold, which I do have a example of that I can show. Would have these organisms in there. And then, there's also different products that I can go through as well. But ideal, the standard model here's, the, um, basic model for why we need these organisms, which is, taught by Elaine, the soil food web. Is, we want to have bacteria, we want to have fungi, which is, all these, let's see if all these, of course, I don't find any. Like this here, this is a piece of fungi. But they can come in
Basically,
all kinds of shapes and sizes. Like here's fungi, shapes, sizes, all kinds of different colors. They can be clear, brown. The classic way of thinking is that the brown pieces of fungi are the beneficial, the most beneficial. But really, it's just fungi in general. You want to have that in your soil. It's very beneficial. So you have the decomposers, the bacteria, and the fungi, and then you have the predators of those, which are the protozoa and nematodes. And protozoa are so diverse. There's so many different genuses and types. The two main types are ciliates and amoeba. And okay, here's a perfect example of an amoeba right now. This is an Arcella amoeba. So they eat the bacteria and the fungi, and protozoa primarily eat bacteria. Nematodes also, eat bacteria, or they can also eat fungi. Microarthropods, isopods, worms, those also contribute to the soil food web as well by eating the bacteria and the fungi. So when the predators of the bacteria and fungi eat the decomposers, then they are releasing plant available forms of nutrients. So this is, the concept of nutrient cycling, and this is, the Elaine school of thought where how soils are maintaining fertility without the use of synthetic fertilizers. So, okay, here's something cool. This is a euglena. This is a mixed trophic protus, and it's actually, you can see it has chlorophyll in it, so it can photosynthesize, and it also can feed off organic matter. So it's like, are you a plant or are you an animal or organism? So that one's cool. So anyways, that's the the standard method of why we want to have this biology in the soil. But as I was explaining last time, there's like so much more to it. Now that we're starting to like understand, there's this is the standard model, and this nutrient cycling is absolutely happening. But there's so many other levels to it, and there's so many other ways that so many other reasons why microorganisms are important to the soil, and so many other functions that they have that contribute to plant health. So.
So having the diversity of organisms
Mhm.
starts with the diversity of bacteria and fungi. And then the diversity of protozoa and nematodes, which
Mhm.
predate on those things,
Mhm.
is what fuels the nutrient cycle.
Right,
Okay.
exactly.
So, I mean, it's kind of amazing to think about, and this is the importance of looking for these things in your healthy soils
Mhm.
and now it makes a lot of sense, hopefully, for a lot of people, you know. It's
Right.
not just that we're looking for these things, but that's why we're looking for these things.
Yep.
Why is it so important to have like a fungal dominance? Can you explain
Um,
that?
you
Or
know,
like maybe
so
not dominance, but fungal presence.
right, right. So, um, yeah, so we'll look at this picture here. So fungi are, you know, really, really important to um creating like carbon in the soil. And okay, so this is a picture of mycorrhizal fungi under epifluorescence. Um, so this is a technique that Matt Powers has really been diving into with his class. So in the past, you know, they the standard method was you had to get all these dyes. So when I first got an epifluorescence microscope, I bought all these dyes and things to try and see mycorrhizal fungi under the microscope. And you found out, or Matt Powers found out, that you don't need that at all. So this is a picture of mycorrhizal fungi, and they are autofluorescent, so they just glow under certain wavelengths of light. So what we're looking at here, these like green outlines. So we're looking at a root, a plant root, and where it is lighting up green, these like really here I have another picture. It's like really lime green spots here that are kind of outlining the cell walls of the plant roots. That's where the mycorrhizal fungi is active. So they're autofluorescent when they're actively transporting phosphorus. Um,
Such a cool display of the network.
mhm. Yeah, so mycorrhizal fungi are, I think they're gonna, it's already known that they are, really important to the health of a plant, but I think it's kind of maybe underestimated at this point as to how, important they are to the plant actually obtaining nutrients from the soil. So that's something I've been diving into lately is measuring mycorrhizal colonization on the roots and, comparing that to the plant health itself in different ecosystems or in different like soil ecosystems. So,
Here's a crazy question, but does
mhm,
mycorrhizae all look like from one root, like let's say a cannabis root to
mhm,
a tomato root? Would it look pretty much the same under what's it called?
epifluorescence. Yep.
Epifluorescence.
Yeah. From what I've seen, yes. Um, it will just kind of like you can't really like see it, but you can see that it is active, if that makes sense. So you can see that it is transporting phosphorus by the fact that it is glowing here. So it is has an active um, relationship with the mycorrhizal fungi. Um, so yeah.
So what's a good way, you know,
Hmm.
so that sample that you were showing us, where did that come? You said that was cedar leaf mold, basically, essentially just collected from the wild, right?
Yep, yep, yep. And then we have this is birch leaf mold. Um, So, this is, basically just, the leaf mold that you're grabbing if you're making a JLF or, something like that. So that's, you asked that question before, how do I get these organisms in there? This right here, this is just like, a tiny little gram of leaf mold soil, and you have all of these beautiful pieces of fungi. Um, I believe, yep, there's nematodes in this sample. There's usually a lot of nematodes in leaf mold too. There's, just so much activity in that first layer of soil.
So just tossing that into a compost tea.
Right, exactly.
Right, just literally because I think that there is a concern. We talked with Ross, and we talked with Ross a long time ago
Mhm,
at Haifa
mhm,
about like isopod issues, gray isopod issues.
mhm, mhm,
So there's this concern, and there should be a level of concern about bringing in raw material from outside.
mhm,
Something like leaf mold, like exactly what you're showing us right
mhm,
here. But by running it through a process, which could be putting it into a compost tea, you eliminate the issues, which to me, I think the only issue would, you could probably list a dozen issues with just bringing something like a handful of leaf mold into the grow at a commercial level. But
mhm,
the biggest concern, I think, at like in a small micro grow is that you're going to be introducing gray isopods, which over a long period of time will absolutely make an impact. So, like, just a simple process of being able to take the good, like a tea
mhm,
or an extract, is crucial.
mhm.
So yeah. Anyways, really, really awesome sample there.
Yeah, yeah, real quick on the gray isopods, we found out that duct tape, they like love duct tape and they like just are really attracted to it and then they just get stuck to it. So if you have problems with those, duct tape works really well.
Are you lining the outsides of the beds?
Yeah. Yeah.
Heck yeah. There's
And
a little bar, folks. People ask
yeah,
me about
there
grays
you
all
go.
the time.
With double sided, or how are you kind of like setting that up so that they
Um,
stick?
I guess you could do double sided, but we don't. We just kind of duct tape it to like the bed is here and then like the duct tape is like this and then they kind of just like crawl up the side of the bed and then get stuck to the duct tape, but it seems like they like the duct tape or something, like they like are attracted to it or like want to eat it or something. So it's interesting.
And I
Yeah.
wonder if
Another interesting thing is that we found that we added like sand and clay to some of the beds at a certain point, just like to experiment. And we found that the beds that had sand and clay in them had less gray isopod issues too. So that's just
uh,
another thing, with soil health is it's not just about the biology, it's also about, the chemistry and like the physical structure of the soil too. So like what it's made out of.
totally.
Mhm.
I also wonder, man, don't know why I didn't ask Chris about this when he was on, Chris Trump, but when
Mhm.
he was talking about IPMO, I
Yeah.
wonder I wonder if gray isopods could be tossed into that mix to find a predatory against them, because
Yeah.
it's an answer to stink bugs. It's an answer to grasshoppers. It's like what
Yeah.
what? Yeah. So I don't know. Just
I'm
a thought.
sure I'm sure you could. I a long time ago found this like recipe to like make a parasitic nematode for like slugs. And you basically just like ferment the slugs in water for, I don't know, a few days or something. And I didn't try it with the slugs. I did try it with the isopods. I did not see any results with it, but I only tried it one time. So something to think about. But I'm sure it would work with the IPMO. That's a really cool technique. Um,
For
I got
sure.
to talk about it this summer. So, yeah, definitely something to try.
Yeah, so we talked a lot about like compost teas and stuff. Can you
Mhm.
show us what a good compost tea
Yeah,
looks like before you apply it? Because
for
I
sure.
think I think that it's important to know that the process that you've put this through like works. If you have a scope and you're looking at the sample before it goes in, it'd be
Mhm.
nice to know like we had a good success or it was
Mhm.
I can do better. So
Sure,
yeah, what
sure.
does that look like?
So
Hey,
I
just real quick, Alexa, are you able to make your screen a little bit larger?
yep,
Yeah, there we go. Boom.
um okay, so here. This is what I'm mainly looking for. Like, okay, this is fungal hyphae here. These white strands. I basically want to see fungal hyphae throughout all of the organic matter aggregates in the sample. So if that's my goal. If I see that, like stuff like this throughout, connecting every aggregate, that is to me like a really good compost tea. And that's what I aim for. It's taken me like, you know, 4 years to figure out, these recipes to get growth like this. And you also want to start with a really good compost too. You can't just, start with any old compost and, expect this kind of fungal growth. So, or the leaf mold, works really well if you don't have access to good compost.
Do you
But
find do you find like anything when you scope? Because I think you said you had. I don't know if we just looked at a JLF or not, but like when
Mhm.
you do a process like a JLF versus a compost tea or like an
Mhm.
aerated compost tea,
Mhm.
like what kind of difference are you seeing in fungal growth like this?
Yeah, so this is an aerated compost tea. I will say that fungi typically grow a lot better in aerated compost teas. They can grow in like a static compost tea or a JLF, but it's not going to be like this explosive, like you're seeing here. So, um, yeah, I typically go for like my usually go-to recipe is like a 24 to 48-hour aerated compost tea. And I dove really into like trying to make the most diverse compost tea I could because like I said, when we started Haifa, I just saw such incredible results from just applying compost teas. I was like, whoa, like what is going on here? But now we know that, a large part of the compost tea is the dissolved organic matter that's in there and how that's forming stable carbon and becoming long or it's becoming mineral associated organic matter, which is releasing nutrients slowly over time. So, um,
So what makes a mineral associated organic matter?
so that's dissolved organic matter. And, another thing to think about with your inputs is, you know, from what I see, anyways, is like most of this stuff, like just because I apply a compost tea that has this much fungi in it, like that doesn't necessarily translate to the soil. A lot of it's going to just die, but that's not a bad thing because, like we know now, soil organic matter is 50% just microbial necromass, which is their dead bodies. And that forms mineral associated organic matter, which is the second most stable form of carbon after biochar. So, biochar is the most recalcitrant or stable, and then there's mineral associated organic matter, which is the second most stable form of carbon. Um, and carbon is I feel like it's something that people, talk about. Like everyone knows, like we need carbon, for soil health, and um, I feel like it's kind of like this thing that people don't understand like exactly why we need it, but it's like the backbone to the soil. It's like the foundation to the soil. So like when you have an environment that has, that's high in carbon, as far as like the soil, then it's going to like set the foundation for the conditions to be correct for like life to thrive in that environment. So, it's creating um, fertility, it's helping to create good soil structure. It's, a home for the microorganisms. So that's why, carbon is important. And that's why we need to, everyone's saying like, oh, we need to add carbon and we need to have stable forms of carbon. And so that's why. But yeah, so the compost teas are great for actually getting that dissolved organic matter in the soil, and then from there, dissolved organic matter can easily be taken up by microorganisms. The microorganisms die, and their necromass can bind with minerals, forming that mineral associated organic matter. Or the other pathway for that is the dissolved organic matter itself can just bind with minerals in the soil, forming that stable form of carbon again. So that's kind of the explanation as to what I was talking about last time. Hopefully,
Alexa, real quick,
way.
for like a layman, would you co-sign that carbon is more of like the currency of the soil food web? It's the money that we're trying to mine and distribute among the diversity.
Yeah, I mean, I guess you could look at it like that. I like to think of it as like the structure, like the foundation, like the thing that keeps everything in the correct order for life to happen. You know what I mean? Like the foundation of a house or something. But that's just like my way of thinking about it. I'm sure that that is correct in some sense too. However,
Rick, do you don't happen to have, Rick Easley was wondering what a good IMO 1 collection looks like. Do you happen to have one of those?
no, I don't.
Okay.
But I'm always happy to
Send
any
one her way.
example.
We'll get her back for part 3.
I'm happy to do it. Okay, so this is a protozoan compost tea. So these are all ciliates in here, and the original like school of thought again, with Elaine's way is that if you see a lot of ciliates, then it's anaerobic and it's, going to cause pathogenic conditions in the soil. But, I there's so many different conditions that cause pathogens. It's not just like aerobic or anaerobic, as I was kind of saying last time. There's a lot more to it. Not saying you shouldn't be careful, because you should. There's a lot of toxic environments out there, but this is something that I apply to my plants. I've applied it undiluted and diluted. I haven't had problems, with pathogens. One thing is though that I have burnt plants because, like I said, a lot of this stuff is going to die when you apply it to the soil. And that can release like a lot of nutrients into the soil really quickly. So, you can use it as like a biofertilizer when you are growing a lot of organisms like this.
Alexa, there seems to be noticeably less fungi connections and hyphae in there.
Yep, exactly. Yeah, good observation. So this is a protozoan tea, and you can make them aerated. You'll just brew it for a longer period of time, like 72 plus hours. Um, I like to do them not aerated. So you basically just make a compost tea, but you don't aerate it. You just let it sit. Usually about 3 days in the summer, 5 to 10 days in cooler temperatures, and you'll get good protozoan growth. One thing that I've noticed with making protozoan teas is if you don't have a microscope, obviously you won't be able to know if the protozoa are growing or when they're blooming, because they do bloom, you know what I mean? Like this, where they're really, really active and then they'll start to die off over time. So to not miss the bloom, I've noticed that if you measure the ppms of it and they go up 150, if it goes up 150 from what you originally had it at when you first made the compost tea, usually the protozoan are blooming them. Then not always, but that's a good indicator. That's the best indicator that I've found anyways. A lot of people say like the bubbles, that can be, but I've found that the ppms are more accurate.
Alexa, you might have already mentioned it, but like, what's the application for protozoan tea? Like, what are
Mhm.
you seeing in your soil that? Because it's also important to understand that what Alexa does is very unique, right? There's not a lot of people that are running indoor regenerative ag, right? I mean, like indoor no-till, I guess would be the right term. Or whatever whatever you box you want to put it into, but having to steer a closed loop is a bit harder. I feel like maybe not, but it might be a bit harder when you're not connected to the ground, when you're not connected to nature, and you're not connected to the bigger picture.
Yeah,
So I would assume that a protozoan tea would apply more into an indoor, like a closed loop setting. Maybe
sure.
not, again, maybe not, but like, when are you going to apply this?
Yeah, no, that's a great question. So I actually I would say, like, honestly, the best application for protozoan teas are like to prepare your soil. So, like, you could apply this before planting, to start cycling nutrients, and just like making things more available. I've also applied it to like compost to help it decompose faster. Um, you can use it as like a biofertilizer. Like I said, you can use it on live plant applications, but I would always dilute it. um, for like, that agricultural soil that we first looked at, how there is just bacteria. One way that we can start to remineralize the soil is to apply a protozoan tea to it, because the minerals in the soil are really important for the biology too. You want to, with a going into an agricultural soil where there's, it's really depleted environment, you could pour that really nice compost tea on there, but again, a lot of those organisms are not really going to stick around. So if we can put this protozoan tea on there and start cycling nutrients, the microbes, that's what they feed off of as well. So we really want to, focus on remineralizing our soil too when we're building soils. That's something that I've found to be like really important. um they're in Elaine's classes, she teaches like, oh, the biology, that's all you need. Like, and I've just found that, you want to have, yes, you want to have good biology, but you also need to look at the minerals in the soil and make sure those are in balance, because I've seen, when the soil has good fertility and good minerals in there, then that also translates to increased biodiversity. Um, Also, you want to have good soil structure as well. That contributes to good microbial biodiversity as well. So that can look like, having different components in your soil, like the sand and the clay. Also could be plant roots as well. So having a diversity of plant roots is super, super important for translating into having increased biodiversity in the soil. So I like to call that like the trinity of soil health. Like biology, you want to have good biology, good physical, like physics, good physical structure, and then also good chemistry. So all those things, we look at soil holistically, all those things together are working together, are ultimately going to give you the healthiest soil ecosystem. It's not just like the biology working as one. You can just add compost with good biology to your soil and that's going to work. Well, no, not necessarily all the time. You have to look at all these other components to really, understand the health of your soil.
So, for anybody listening, so we have 45 on YouTube and 9 on Instagram, which is it's a far cry from the amount of people that will be watching it in the weeks to come, and I think I'm hoping years to come. But so for anybody listening right now, though, and if you're listening in the future, remember we'll be here every Friday night at 9 p.m. live. This is a Q&A, guys. So if you have questions, and I think that when we have amazing guests on, the questions sometimes come a lot later because there's so much information, so much good knowledge being dropped. So hopefully you guys can drop in because we are looking to connect with you guys. But I do have a question, and it pertains to predatory nematodes. So you're commercially available, like your H-back, your Steinernema feltiae, your Steinernema carbocapsae.
Sure.
So my understanding on those is that they're a indiscriminate killer, so to speak. Or in like a lab study, there's been tests done where like your Steinernema carbocapsae will parasitize or use a host on, like some close to 50% of any host in the culture. Don't know what the parameters was of the study, but the question I have is, since you guys have probably used these predatories, it's not, from the last time we spoke, you spoke about them a little bit, so I would assume that you've used them before. Do you see any diminishing return on using those in a closed loop system? Like, are they attacking beneficial microorganisms on top of what you're actually after? Does that make sense?
I'm not sure. But if they are, it's a part of life. You know what I mean? I think we do use them. And they do persist. I've seen them persist in the soil for, about 2 weeks. After that, I really see their populations start to drop off. But we use them. And, I think, I don't know, if there's other kinds, but the kinds that we get and that I've, heard of, mostly just infects like, pests. But yeah, I guess so. They infect. You're saying that they are infecting like tails and other things.
I guess, yeah. Like my concern, right, with so the product that we offer is our biodiversity pack, which is rove beetles. It's going to be springtails, predatory hypoaspis miles or stratiolalips. And then like soil mites and stuff, which would be more like, to me, those are going to be like your feeder mites, right? Like that's your bacteria for the protozoa in the system.
Gotcha.
So to me, the only concern that I have in a closed loop system with adding, so like if the attack is on fungus gnats,
Mm-hmm.
there's going to be collateral damage with the other beneficials, because those beneficials, rove beetles do eat like potworms and
Yeah.
smaller microorganisms, but they don't eat they eat the bigger microorganisms. They don't eat the small microorganisms like those predatories. So
Right.
to me, it conflicts with the bigger organism attack, for fungus gnats specifically. And then like
Mm-hmm.
as far as like ag and outdoor using them for other pests, I couldn't really speak on that. But
Sure.
that's it. That's kind of where the question comes from, is like, are they going to attack, are they going to interrupt the rest of the system? Because we talk about the wave of nature, right?
Mm-hmm.
And I don't know that those parasitic nematodes exist in such concentration in
Right.
nature, right? So that's almost like interrupting the wave of healthy soil.
Yeah.
So I don't know.
Yeah,
Thinking out loud.
yeah, for sure. With our soil, like you said, it's not necessarily like nature, you know what I mean? We try and mimic nature, but it's indoors and it's constructed by us. So, we have to kind of do certain things
Play
to
God. You have to play God. Yeah.
mm-hmm.
Yeah.
So for us, we don't necessarily see it, harm the beneficial populations. I think it just keeps everything in check. You know what I mean? It doesn't even get all the fungus gnats and stuff like that. It all works together.
Those are kind of part of the system in small
Right, exactly.
concentration as well. Yeah.
Exactly. Everything kind of has a part, even the things that we see as like pests or pathogens, they're playing a part in the whole picture. So,
Hey, Alexa, I have a question.
Yeah.
We probably start getting onto these questions too, but my question is there a let's say I take my compost. Is there a way to steer my compost tea towards fungi or towards protozoa simply through the process of cooking it, boiling, steeping it, whatever the case may be?
Sure.
There's more air applied. Are we going a certain direction as opposed to less air? Can you help me
Yeah.
with that?
So, like, the fungi grow best in aerated compost teas. Protozoa can also grow really well in aerated compost teas. But they can also grow in the static compost teas. But I think the biggest mistake I see with people making compost tea is just simply adding too much food or source. So you really don't want to add too much. Like the amounts I add, it's like 1 ml per gallon, 2 mls per gallon, of the food or source. And that's all you need to grow really good biology, because I've tried recipes that use more, and all you're really gonna grow with like more food resources is bacteria. You'll find a balance to where you're growing these organisms um, with just smaller amounts of food resource. So that's the one thing that I see people doing, is just really adding too much food. And then I will say, like, my like go-to recipe is fish amino acid, 2 mls per gallon, a sugar source, or you can do any fish source. So I'll just say like 2 mls per gallon of a fish source. I use fish amino acid. You can also use like fish hydroxylate, or what's that other one? Can't remember. And then you can also, and then so 2 mls of fish, 2 mls of a sugar source. So you could use like a fermented fruit juice or molasses. I like both of those. And then 2 mils of a mineral source. So that could be like kelp or rock dust or, I don't know, C90, something like that. So that's like my base recipe. And you can just off different brewing methods grow more fungi or more protozoa with that same recipe, as long as your inoculant has those organisms in there. If your inoculant doesn't have those organisms in there, you're not going to grow the organisms, obviously. So
Seawater work as a mineral source?
yeah, I'm sure that would work. Yeah. So that's what I do. And that's what I've seen good results with. Let's see.
So Brian, this is probably a great time because we do actually have like 10 questions.
Oh wow, really?
Yeah,
So,
they pop up for you, honestly. So I know you're always apologizing, saying you don't know this or something. You don't. People resonate with you, and you should know that.
Oh, thank you so much. Yeah, I appreciate all the comments on the last one because I felt like I did so bad, and then I read all the comments. They're so nice. I was like, oh.
No, yeah, no. It's hard to even cut these clips to kind of promote the show because I don't like listening. It's just inherent. You don't like listening to yourself. So don't ever think that you're crushing. Your information is invaluable. Truly, there's been a lot of gold bars in this episode.
Okay.
And another Quatamok was another episode that you have to go back and listen to.
Yeah.
Same with this one, guys. Unfortunately, you're gonna have to go back and listen to it and make sure you smash the like button because we really appreciate it. Definitely seeing the subscribers go up, and it gives us fire to keep moving on this show and keep promoting our friends in the community. Man, this is amazing. All right, Brian, let's get into them questions.
All right. A lot of regulars, Alexa, asking some questions tonight. So thank you, Guy Grows High. When scoping a tea, should one collect a sample of foam or the liquid? Is there a protocol for tea that differs from scoping soil?
That's a good question.
Great question. So yes, I don't dilute compost teas, but what I do is, okay, something super important about compost tea is pretty much the like particulate matter that is in your compost tea, it always kind of settles at the bottom. That's where all the organisms are. So I'm a super like freak about mixing my compost teas when I'm applying them because like all of the organisms are, not all of them I should say, but a lot of the organisms are going to be within that like little particulate organic matter that kind of like settles at the bottom. So what I do is I collect some of that like silty stuff, the stuff that settles at the bottom. I'll take that and you'll see that that liquid is a lot darker. And then I'll take some from the top, where it's not as dark, where there's less particulate material, and I'll put 1 drop of each under on the slide, cover it with a cover slip and look at it that way. So I kind of think of that as being like a good ratio of kind of like what you're getting. So when you take from the top, that's primarily just bacteria. There's not a lot of activity in there, but that stuff at the bottom. Do you guys know what I'm talking about? Like with your compost teas?
Oh
like,
yeah. I just did this.
I
did this the other day. Put a sack of compost in a 5-gallon bucket, and I let it set for like 2 weeks. Just sit there. It was after your show because you said something like this, and when I pulled it out and I drained a little bit of that top, it was like looking at gold particles down there. It was so fine, super fine. It was great.
Yep, that's the good stuff right there. So always like makes your compost teas really good to distribute that stuff evenly when you're applying it, because that's where all the good stuff is in that little particulate material.
All right, second question from David Graff. What's a good starter microscope? I know last time you were here, you were talking about some of those.
Yeah.
But as Mark just had mentioned, the show is really starting to take off. So we
Mhm.
appreciate each and everybody that is here on Friday nights, and this is starting to become a fun little powwow. So what is a good starter microscope? I would say starter microscope for some is probably 150, and maybe for others around like probably 500. So if you could kind of do both of those price points, or if it's you know more about them than I do, maybe it's a little bit cheaper than 500.
Sure. So for starter microscope, I would check out LW Scientific. They have really good like promotional deals quite a bit, so just like keep your eye out for those. Um, and that's what I got. That's what I have. Um, you can also, look for like sales. A lot of the times, people will say they, found a microscope from like schools, giving them away. Like I said last time, you can do it like that as well if you can find, people just like giving them away in your community, and offering like that. Um, just look for a shadowing condenser. That's the main thing you want to look for when you are doing soil microscopy, because the organisms are actually like the same density as water. So you're actually looking at their shadow. So you want to have a shadowing condenser in order to do that.
And then there's kind of like a follow up. The community was answering a little bit here about kind of the norms, to be honest. And then Jeff Lowenfels, Dr. Lane Ingham. Is there a few others that you would kind of put into that I know that you're on a deeper level than some of the like major names that people have heard? So maybe some less known names of books that you would recommend?
Mm, honestly, like Matt Powers' new book, I feel like it takes you through the basics, but it's also like a deep dive as well. So like it's kind of like everything you need to know to like get started, and also like more advanced things about soil microscopy. So I that's like the number one book I would recommend for people, especially just getting started. It's really comprehensive.
Yeah, almost any of those books
Yeah.
overwhelm me.
For real.
Do
For
you read
real.
journals or anything like that? Academic journals, white papers, things like that? Do you get that deep? Like
So
read?
like the the stuff where I'm talking about like dissolved organic matter and mineral associated organic matter, those are journals as well. But like a lot of the stuff that some of the stuff that I read is not like public access, you know what I mean? So you have to like get it through a institution or
Yeah.
like someone that that has access to it. Unfortunately, so that's just the
You can probably buy each
like,
and every individual one, but you have to have like an .edu email usually to have access to stuff like this.
yeah,
Back when I
exactly.
was in school, I could access all those institutes and all of their papers. It was fantastic, and I can't do it
Yeah.
anymore.
And I never did anything with that power when I was in college. Fast forward 15 years, here we are. Wishing I had it. Unbelievable.
Right. Exactly. But you can just search, different keywords like dissolved organic matter, mineral associated organic matter, microbial necromass, whatever you're interested in, you can search it. There are free ones. I would just recommend doing that and seeing, what's free and whatever you're interested in or your questions are specifically. So
Yeah, we had one from Hungry Plants here. I wonder why the mycorrhizae makes biophotons.
hmm, yeah, I guess I don't know exactly how that happens. I don't know.
Is that why it shows up on the epifluorescence? Is that where
Um,
the questions? Is that
yeah,
what we're talking about? This
this is a
goes over my
question
head.
for Matt Powers. Really, you'll have to ask him next
Hey,
week.
Hungry Plant. Next week we got Matt Powers coming
Exactly.
on. So yeah,
I
drop this
don't
question
know
next week.
exactly how that works. That's what he explains is that the phosphorus within the mycorrhizae, when it's like actively transporting it autofluoresces. I think it's autofluorescence rather than like biophotons. So you know how like some like fungi or like I think there's like also like some bugs that autofluoresce. They just like glow. Um, I think it's something more like that. So under certain wavelengths of light like that efflorescence, this specific the mycorrhizae just autofluoresces, so it glows under those specific conditions. That's my understanding of
That's a
it.
pretty good explanation.
Okay. Yeah, but check with Matt Powers on the correctness of that.
This is a question for me, Alexa. I was able to kind of go around, I think it was like 2019 and 2020, early 2020. And it seemed like a lot more of the living soil growers were adding green sand and then getting away from a lot of the diversity in the cover crops and just focused on grasses from a labor standpoint. Since you are managing a facility of relatively decent size for the how you guys don't have a bunch of suits and all that kind of stuff. It's a huge operation, in my opinion, for the small time person. So thinking long term with that team, that little, the small but mighty type team, is green sand in your tool belt? And what are your thoughts on growing the diversity in the cover crops, or just moving to the grasses to keep it easy?
I would not. We tried the grasses to keep it easy, and it did not work for us. Definitely threw off the balance of the ecosystem. It attracted a lot of pests, and it did, had detrimental effects on the microbiology as well. So diversity is really key as far as cover crops go. We made that mistake.
So it's a shortcut, but it's a shortcut to potential issues. And
Yeah.
then you want to go more to like in my opinion, I guess the most famous one would be like the Build a Soils 12 cover seed blender. They're always messing around with how many seeds they got in there. But.
Yeah, we started with that, but you'll kind of, pick out what works for your system. So there are certain things that in our system attracted pests we had to get rid of. And then we kind of like added certain things. And
What kind of pests were you pulling in? Like mites?
yeah, all kinds of stuff. Mites,
Yeah.
root aphids, thrips, yeah.
They just like seemingly materialize on the
Yes,
it's unbelievable how that works. True,
crazy. And then
it does happen.
they go away. Like it's so crazy. People are like, "How'd you get rid of the root aphids?" I'm like, "We just got rid of this cover crop." They're like, "What?" You didn't have to.
So what's the secret sauce then? Like
Yeah.
what's your fallback? Like what's your go-to?
So we typically, just use dichondra and buckwheat. And then we also use like all those different like herbs and perennials like tulsi, rosemary, basils, different things like that. Herbs like culinary herbs.
You just do that because you use them in the kitchen too, huh?
It's
It's like it's amazing double-edged sword.
yeah,
And gold nugget.
balm is really great. Mints are really great. Yeah, they just don't like really attract pests for us. So
Yeah, we
but
used to. when I was single, we would grow the chocolate mint and then have mojitos, like when the ladies come over and just pick them out of the fresh. That was a little slick little move back in the day. You know? Oh, you like to garden? Here's some freshy fresh. All right, this guy, I think what we saw earlier, this guy's kind of been acting out of line or whatever, so I don't know how you want to, but he did ask a question, so I ain't going to give him that, but I think he was acting kind of stupid. So Mr. Rusty Nails, don't do this shit anymore. What am I getting by just using castings in a short soak extract rather than a bubble tea?
Okay, yeah. So in an in an aerated compost tea, like you will get more diversity. You'll also get more fungal growth. But if you just short soak, I'm assuming that's probably more of like he means more of like an extract. So a compost extract is when you're taking your compost, you have it in the compost bag, and then you're running your water through it. You're catching the water. The water as the water is running through it, you're massaging the compost to release that dissolved organic matter, the microorganisms, the humic acids, minerals. So that's going to be released into the water, and that does have more diversity in it technically before you brew it, but it has a lot less activity. So it kind of depends what you're going for. Like if I'm pressed for time, I will do a extract. It also takes more compost to like make a compost extract. So if you don't have like access to a lot of compost, I always recommend making compost teas because you need a lot less compost to make the same amount of organisms, if that makes sense. So compost aerated or brewed compost teas are gonna have like exponentially more activity in them, not necessarily more diversity, but when you nail down the teas, you'll have really good diversity to where it's like, um, I usually make compost teas now, but I'm kind of getting back into making more compost extracts too because of the dissolved organic matter. Knowing that now, compost extracts, you will because you use more compost to get those. Typically, you're gonna have more dissolved organic matter as well.
Yeah, because the tea is it's not gonna breed dissolved organic matter.
Right, exactly.
It is or it isn't.
Yep.
Yeah, but it's a good way to get it in concentration,
Yeah.
and to be able to spread it across a playing field evenly. Yeah,
Yeah.
yeah, which is
Exactly.
powerful tool. Another tool in the tool belt.
Mm-hmm.
That liquid gets in the soil quick.
Yeah, exactly. So they're both really good for different things. I would recommend, using both. You want to use some aerated compost teas and some compost extracts. They have different benefits. They're both really beneficial. So, 1 week use your compost extract and then next week use your compost tea, something like that.
For people that are newer to this and maybe don't have enough money or don't want to put enough money into it, in the early days we were doing this like dunk and squeeze. We were using our girlfriends or our wives' pantyhose and usually using like a stick or a wooden spoon and kind of keeping it on there and then just squeezing it, bringing it up, trying to aerate as best you could with your own hands and that kind of stuff. Do you feel like that's still going to be something, or is that bro science? You actually need to add the oxygen.
No. So yes and no. So that could be something. I will say when you really need that 400 microns openings to get the microorganisms out of there. So you'll be getting some bacteria and you'll be getting dissolved organic matter, which is really important. So you'll be getting those two things with that method, but you won't be getting like your protozoa and your fungi and like your nematodes and larger organisms. So it just depends what you're going for. Like that method, if you need something simple, there are benefits in there. But if you're going for like, hey, I need more fungi in my soil, you'll want to go towards like a aerated compost tea, because that's really the best way to grow fungi quickly for your soils.
So obviously you can just build up to it. If you don't have the money, then just kind of
Yeah,
do it that way, and then once you got it,
yeah.
get your little bucket or something.
And like what I do is like you don't really need like an expensive brewer or anything, although I'm sure those vortex brewers are really nice for growing microorganisms. What I do is I just have like two pumps with tubes on them, and then I have them connected to a like weight at the bottom. You can use a rock. I use a pickle fork. And you just have that at the bottom of the compost or a 30-gallon trash can or something like that, and just bubble it that way. And that's how I make all my compost teas. You don't need like a fancy brewer or anything, just like a couple air pumps. Those are the most expensive thing.
Bars right there. We used to be able to do the vortex. We would cut the
Mhm.
big blue, like I think it's usually 5 gallons of water. You just flip that over, cut it in half, and then run the plumbing, and you can get that vortex going with the proper pump. So you don't have to spend a lot of money. I think it maybe was like 150 bucks back in the day. So now it's probably $1,000 with the craziness of what everything costs.
For sure. Yeah, no, I gotta make one of those.
We can do these last ones pretty quick here. They're kind of all just still about the compost. A quick question
Okay.
from Rick Easley. What's the bigger difference between compost tea and worm tea? And I think he means like running it through, obviously, with like the red wigglers and all that.
Mm. So like the worm tea, like that, like leachate.
I think he mean no. I don't know that. I don't know that he's asking about like the stuff that you would like pour off the bottom of one of those.
Okay.
I think
So,
maybe he's wondering like using worm castings
okay, actually,
versus
yeah.
like live, like your best cedar
Mhm.
leaf leaf
Sure.
mold.
So this is a good question, because I always recommend that people use their worm castings for making compost teas, um, because it's more, sustainable. So like I always recommend for people to or people who don't have like access to forests, really close to them, keep a worm bin, and just put like a carbon source in there. So just like the leaves work best for the fungi, because the worm castings themselves don't like really harbor any fungi. So you want to put a food resource for the fungi to grow in there. So if you just add like some leaves to your worm bin, that is a really great way to make an inoculant for compost teas. I do that as well. Um, you know, I do add like leaf mold from the forest to my worm bin to get the fungi out of there. But worms have such a diverse digestive tract, so there's like nematodes and protozoa and all kinds of really beneficial bacteria in there. So I primarily make most of my compost teas from my like worm bin, that I guess I call like my mother culture. It's like a like what the
Totally, couldn't agree with that term.
for for
Like a
for
SCOBY.
yeah, like a SCOBY or like a sourdough starter, except for like
Yeah,
compost teas. So
compost starter.
yeah,
That's
compost
that's it.
starter.
That's essentially what our biodiversity packs are. Is it
Yeah.
just? It's just it's a huge hit starter pack to get shit rolling.
Nothing
And
better.
that's a bar right there, because having it can't be said enough that having an at home worm bin will pay dividends. And what Alexa just said is you start the worm bin now, years from now, this thing, every handful is going to be like gold to start a new worm bin. Literally. It's just time, guys. Start it now. So huge bar right there. And then what have you looked at, like, or have you ever thought about using the leachate or leche or whatever, the pea coming off the bottom of the bin? Like, what is that?
My worm bin, I don't get any of that. I just have it in a tote, and do it that way. I'll
Yeah.
do the method where you just put the food to one side and they all go over there, and then you harvest the other side, and then you just kind of like refresh it that way. So that's the method that I use. But I'm sure you could use that leachate stuff. I don't know. I just don't know because I've never used
Just thought
it.
I'd throw it in there.
No
No worries. Yeah, no worries. You can answer the question at hand very well. So
Is compost tea better than compost starting?
I think what the question is using a compost tea better than putting compost on your garden? And please clarify if you mean different than that.
Like a top dress.
Like a top dress to the garden. What's better?
Okay, so I just read this study that says that, this was a really big shock to me because I was like, oh, like I love adding compost, but 60 to 90% of the compost that you top dress on your soil is not actually like making it down into the soil and becoming like a long-term form. A lot of the nutrition that's in there is getting eaten by microbes very quickly and then respired into the atmosphere. So they're saying that like 60 to 90% of the stuff that you top dress, or the organic matter that's top dressed, not just compost, like any organic matter that's top dressed is just going into the atmosphere. But the study also showed that if you took away that organic matter layer, it had a detrimental effect on the formation of like longer term organic matter in the soil. So it's, you know, if you take it away, it's not good. But it.
Would you say that this is where you, like, what during your let's say like your spring regiment, right? Like let's say for a second you're talking about, well, actually either way.
Mhm.
Let's say it's your spring regiment where you're adding compost from maybe your pile. Like, this is how I do it personally because I run static piles, but I'm adding compost to my garden.
Mhm.
Would this be a time when actually adding some mulch layer to give them a fighting chance to get down in there, right? So maybe again, because it's always understated the importance of that mulch layer, guys.
Mhm.
Again, here's a point, like here's where, like, bringing in some wood chips or bring, like,
Oh,
whatever you have available around you, and giving that a buffer, I think would probably be a good idea.
Oh yeah, absolutely. So that was like one of the main points of the study too, is that like layering stuff on top of the compost and organic matter and things like that. That's gonna help get it into the soil and have those nutrients stay in the system rather than just get like blown off into the atmosphere. So that's really important. um and I got to see that firsthand this summer. One of my friends has a bunch of hugelkultur mounds, and he mulched one of them and also planted it in the syntropic agroforestry style, where he has different trees and vines and herbs and all these different plants in there with his cannabis. And we saw that the layer that he like mulched and planted like that was like heads and tails healthier than all the other hugel mounds. like it didn't have any septoria, and it was like, super lush, super green. So, and it was like really all he did is like mulch and had like diverse plants in there. So, it just speaks to how, you want to treat the soil as a system and really look at it holistically, and just, know that no one part is greater than the whole. You want to like look at everything and consider it all, and don't get too focused on one single aspect. And um, so to answer that previous question, I would say that, you definitely, you want to put compost on the soil. Absolutely. I would never stop just because of that study, saying that it gets blown off, but maybe I would just like add more mulch on top of it. Um, as far as like getting like activating your soil and getting those long-term storage forms of carbon, that's those slow release nutrients, um, definitely doing the compost teas and getting that dissolved organic matter. So when you're picking a compost for your compost tea, you want to make sure that like when you're running your compost, or when you're running the water through your compost tea bag, that you're getting that really nice brown water coming out of there, like deep dark brown water. The more deep dark brown stuff you have in your water, that's like the dissolved organic matter, that's the microorganisms, those are the minerals that you're getting from that material. So that's like, when if you don't have a microscope and you're trying to pick a good compost, that's like one indicator that, okay, this compost is really good, is if you have that. Because I've seen a lot of compost, like industrial compost, where you don't get that when you're making compost extracts. So there's something to be
All
aware
right. Yeah. And this has kind of been fun. I've been managing Instagram, and so sometimes I'm not clicking the buttons fast enough. But this has also been fun for the people that are wanting to get with our very own Mr. Kevin here. And we have a question here for you, Kevin. And then if you guys see at the bottom, there's scrolling there. Kevin is speaking this weekend at a mushroom fest, which is awesome, man. I mean, the public speaking thing is, it's almost like if you're a comedian, you need your stage time because every audience is different. But the way that you guys have set this up, I think it's Jacob DeVecchio. Sorry if I mispronounced that, buddy, but he's the brains behind it. And so they want to know what you're smoking on, what you're bringing, and take a little bit and talk about your talk this weekend as well.
Yeah. So yeah, I'm in Oklahoma City. I'm in a hotel by myself, sitting on a bed. That's why I'm so quiet, man. I've had a few milligrams and we're at a mushroom festival, and it's a freaking fun time. Tomorrow I'm going to be a judge in a cannabis competition. So I got big plans for myself and this hotel bed, man. That's about it. We always giggle about how us three dudes with kids on a Friday night, we're the only people on the internet talking, probably because everybody else is out doing something fun. But when you got lives like ours, you either hustle or you deal with your children. So yeah, man, I'm going to speak on I'm the very last set of there's 3 speakers going on every hour for 2 days straight, man. It's fantastic. And I'm the very last group. I speak at 4:00 on Sunday. And my lecture will be, or presentation is called "Let There Be Soil: Mindset and Methods of Soil Creation." And the theme of the festival is loyal to the soil, which is interesting because it's a mushroom festival. And you might think it'd be loyal to fungi or something like that, but the theme is loyal to the soil. And I was here last year, which was the very first Oklahoma Fungi Festival. And I was asked to come back again this year and to speak this year. And it's very relevant this year because I'm bringing all the IMO 1, 2, 3, 4, and 5, compost, and bokashi. And I have my interactive totes at my table. I'll post a video on Instagram tomorrow, but it's all interactive. They can look at the material, the end product, and kind of at least see how mine looks. And it kind of helps you. The reason is when you can see the end of something, it kind of gives you the ability to move forward faster, or it does for me. because it's like, oh, that's what I'm going after. So now I at least have that example to work with. And there's lots of examples, but at least you have an example that you can touch and feel. And man, this stuff looks good, y'all. I worked hard on it. It looks really good because I've got tons of this stuff, because next week we go to my autumn immersive class in Tahlequah, Oklahoma. But man, this I got to meet Inoculate the World. I don't know if y'all know him on Instagram. He's a big mushroom dude here. Really super nice guy. Okay Mush is here. They've got some beautiful inoculated blocks that have these long horned reishi mushrooms coming out, and all these cool lion's mane mushrooms in all the bags. There's all kinds of liquid cultures, spores, swabs.
Matt Powers.
Matt Powers is going to be there, guys. He's going to be on our show also in what, 2 weeks? Or when is
No,
that?
next Friday, man, my
Friday.
guy.
Oh yeah, that's right. Next Friday. Sorry, guys. Yeah, that's right. So he's going to be speaking also. So I'll get to meet him tomorrow. And guys, my table, you'll see it's a beautiful table. I love it. But I'll have my computer set up and I'll have this show going tomorrow. I'll let everybody. We're going to do the replay, Organic Alexa, and you're going to be on it. Everybody be watching you tomorrow. So go to okmushroomfestival.com and you'll see pictures and fun stuff like that. Let me actually, one more thing. What my lecture is I'm going to take them through the history of how I got to where I am, and then I'm going to tell them what I do to make my soil. So it's kind of 2 sections of mindset, how I got here, what I was thinking, how I changed even, and then go into the methods. Here's what I do. Here's step-by-step things to do to make some of the soil, and remember those mindsets as you do it. And I think it's going to be a good little talk.
No doubt, man. You put love into stuff, so it's going to come off, and then you have the passion. And then whether we like it or not, we're getting a lot of practice on Friday nights compared to other people. So there's a little bit of a polishedness to the way that you guys speak now. Out of respect for Alexa, I'd love to let you go next and kind of highlight the things you do, and then you can fall off and we can just finish and wrap up. I know your time is valuable. But what are some of the things that you're working on in Haifa? I know that people that are close to your area they always kind of want to support the small time farmer. They might not necessarily know about you guys, unfortunately, because of the way even the algorithms work. So if you guys are, just real quick, if you guys are watching this stuff on Instagram, just know that that's almost the worst platform for us. They almost go out of their way to suppress the information that Alexa's giving you tonight. And which is really sad, honestly, because it's free. I mean, I don't know why they would suppress this kind of stuff. So if you follow us on Instagram, you're able to support the show. And all we ask is hit the like and the subscribe. For whatever reason, man, that makes a huge difference in that algorithm. So that's why you hear people constantly talking about it. So Alexa, you're growing a double-decker living soil grow, perpetual grow, which is, as far as I know, not many people are finding success with that. So kind of while you're hyping yourself, do you mind kind of talking about why you guys are doing two floors?
Well, honestly, we did that just because we were trying to, build something that could, produce enough for just like a small grow, like it's a very small grow. So we had limited space and limited money, limited materials to make it work. So that was the reason we went double stack. I wouldn't necessarily recommend it for anyone to do that unless they have to.
It seems so hard. It's almost like spinning plates or something.
Yeah, it was hard to figure out that system for sure. And if I had to do it again, I wouldn't. But but yeah, we made it work. So if you have to do it, you can. But it's just like, just using what you can or what you have available to you to make it work. So
And Alexa, so you also have like a personal website. It's organicalexa.com, correct?
Yeah.
And you offer kind of something special, a qualitative and kind of quantitative soil analysis. So I think it'd be having a Logan Labs type soil test is definitely beneficial, but having a qualitative, meaning somebody giving you the kind of the information that you've been given tonight, but
Mhm.
how to tailor your soil to do the best.
Exactly.
From a more of a microbial standpoint, rather than like
Yeah.
just numbers and figures, minerals and macro micronutrient standpoint. So
Mhm.
to support you, what's the best way to kind of get that process started
Yeah, just
at Organic
go to
Alexa?
my website, organicalexa.com. You can fill out a contact form there. Just tell me like what kind of project you have in mind. I do offer, like you said, soil sample analysis for microbiology. So with that, I'll give you the quantitative analysis, which is like the data of how many organisms you have per gram of soil for each functional group. So bacteria, fungi, protozoa, nematodes. And then I'll also take pictures and videos of your samples so you can like see what's going on and have a better understanding. I also, if you do have a Logan lab sample, I can look at that for you and tell you, kind of what we need to add to remineralize it. So I can do like a full soil health consult where we're looking at, the biology, the chemistry, the structure of your soil, just your practices, and then, provide you with, different tailored compost tea recipes, composting recipes, different recommendations, based off the data to improve your plant health. So,
Sounds awesome. We need to do that with my garden soil, man. I need to take samples from the no-tills and really have this done. They've been going for 6 to 7 years, my old ones, and new ones are under a year old. Yeah,
You
cool.
I need.
got your girl. You got your girl right there.
Well,
Yeah,
yeah,
I'd love to
exactly.
help you with
It's
that.
perfect. Yeah,
What an
I'm
awesome project.
Yeah, I'd love to. It sounds fun.
And
Yeah, no.
if you want her resume, you've got her resume. She runs basically an impossible operation from the biology side of it, right? I'm not saying that you run the whole operation, but she keeps these plants happy and alive in nearly an impossible setting, using regenerative practices, which is absolutely amazing.
Mhm.
But beyond that, real quick, because I feel like we would be failing ourselves if we, as far as Michigan and as far as your Michigan mission. Sorry, I don't know. There was a play there on words, but
Cam compost.
Camp Compost is something that everybody should get behind. It's a regional thing,
Mhm.
and I hope that it's a program and a platform that people can apply elsewhere, because it's something that I beneficial as a company. It would be happy to support. The show is happy to support. And it's something that if you're a small business in regenerative and you're looking to support a mission, please think about supporting these guys as a sponsor. It's really not that expensive to get on the list and to be a part of something amazing. But we talked with Quadamak Villa last week. He was one of your keynote speakers at Camp Compost. We have, if you didn't see that one, God, you got to watch that. Equally as amazing as this episode and as the last episode we had with you, Alexa. In a couple weeks, we have Timothy Hammond, who is a quantum biologist. I guess I don't know if that's an actual thing, but he's a quantum biologist
Mhm.
who has an emphasis in the importance of regenerative agriculture and eating a local and seasonal diet. So it's really amazing. Like the people that you guys have at Camp Compost are the people that we really want to talk to now, and it's like now we're on this parallel.
Yeah,
So
amazing.
like
Keep it going. I
seriously,
love it.
Alexa, it's like it's amazing. But What does camp compost mean to you, real quick? Because I think that we should definitely say that, because you are one of the co-founders and the lifeblood behind this thing. It's amazing.
Yeah, camp compost is like just we created this event so other people and each other, we can all connect with nature and just really, come back to like the soil and, who we are, at the root, and kind of step away from all these, constructs in society and really, you know, just be like rewild ourselves and, remember that we are nature and we're not separate from nature. Um, we are part of her and we need to, work within her cycles. And that's really what we're doing with each of the, amazing presenters, all sharing their unique way of how they work with Mother Nature and incorporate her wisdom into their lives. And, you know, it's just so inspiring to hear all of the incredible ways that we can work with nature. Like Quadamak, he is working with nature in bioremediation. So we got to hear about how he is bioremediating PFAS from the soil. And Tim, who's incredible, he's, like you said, a quantum health practitioner. So he's looking at our light environment and how the different, the sun and the different lighting and even electromagnetic frequencies that we're exposed to on a daily basis is actually impacting our health in different ways that, we can be more in tune with the sun and live in harmony with nature and, improve our health through our light environment as well. His talk was absolutely amazing at Camp Compost. So I'm so excited for like this information to be able to reach more people. I've started applying the techniques into my life and it has been very significant. So I'm glad this information will be reaching more people through your podcast soon. He's incredible. But yeah, Camp Compost really is just like a, heart opening experience. And we would love to, have anyone who is interested join us, join the community. It's just such, a loving group of people with so much knowledge and wisdom. And, everyone is, so giving and sharing and kind and honest. So it's really, just a beautiful experience that we have every year. It's usually we haven't confirmed dates yet, but I believe we're going to keep it in August this year. Just stay up to date with us on Camp Compost Instagram, and our website is campcompost.com. So we'll be having more information as the new year comes in, and we're so excited to plan this year. It's really just been getting better each year. So thank you for that. Yeah.
Truly amazing. I love you guys. It's at Organic Alexa, at Kulianic Consulting,
Yes.
and at Jet House Gardens.
Mm-hmm.
Those are the three co-founders of Camp Compost, and then at Camp Compost. So follow those people. Amazing shining lights in this community. And man, we thank you so much for your time, Alexa.
Mhm.
So I'll sign off, and then Brian can take it away. So mibeneficials.com is how you can support us. I'm stoked because I got my Super Sarah powder and my EM in the mail from Terraganics, and then I was surprised to get a phone call personally from Terraganics production manager to explain how to get the best out of my product, right? So I'm going, wow, I made it to the top, dog. So then fast forward a few days, and I'm like, you know what, I'd love to be able to offer EM on MI Beneficial. So I signed up for a wholesale account. Well, one of the owners of the company called to set up, like, to have the initial conversation, and I'm like, hey, do you guys call everybody as like a standard thing, or am I just like special? She's like, no, yeah, we knew we started doing that. Everybody gets a phone call. I'm like, holy crap. So big company run like a small business, truly amazing company. And we have them coming on in a few weeks. They're coming on December 6th, so that's going to be a fun episode to catch up with them and see how to get the most out of those products. But other than that, mibeneficials.com, we have both the rainbow springtails, uh, that hoodie on the site, and then we have the Friends with Benefits hoodies back up on the site. Small drops, local made. We're not running through like a big conglomerate site ready that it just gets made when you order it. These are already made, so they're very limited. And other than that, just really appreciate all the support for the show, and it is an absolute privilege to be able to um, support other small businesses and other entrepreneurs and people that are like-minded in this regenerative world that we're living. I think that we are far ahead. The people listening here and the people that come on these shows are sharing this information. I think we're far ahead on this kind of this snapback, where we see where climate change is taking us, and we have an opportunity to make a difference, if not just in our own backyard. So let's let's take this information and run with it, guys, because it is amazing, and we cannot thank everybody on here enough dropping these questions in. We can't thank you guys enough, because that's really what makes this show amazing, being live here with you guys and being able to make these people accessible. um Brian, what's going on, brother?
Yeah, I mean, besides learning each and every week, there's definitely a formula here where people are finding value in it. So hats off to everybody that's starting to come together and find a little community on a Friday night. For me, I had to rush here to be a part of this, as I think Kevin was behind the scenes, and Mark usually has to do as well with life. And so we're having Repticon this weekend. It's, at least here in Colorado, it's a huge event. It usually takes like a day to set up. So my poor wife is probably setting up as I speak with our young children. So shout out to her setting all that up while I'm here goofing off with you guys and enjoying life. So there's something to, again, just teaching yourself how to do this kind of stuff. Find your own way, and whether that's taking it to like an elite level like Alexa, or maybe just knowing that Mother Nature works, probably like myself, and then finding people like Kevin and Mark, and being able to run with this kind of stuff and find success. So just hats off to you, especially to Kevin. It's cool to see you be a part of something like that, man. And public speaking is usually very scary. So it's cool to see others that aren't willing, or are willing to kind of get up there and just run with it.
Yeah, I appreciate that, man. It's fun for me because I'm just going to talk about things I love to talk about, and it's like I have a captivated audience. They just sit there and have to listen, and it's just me getting to chat and enjoy what I hear of myself saying. It's sad but true. It's just like I love talking about this stuff. New things will pop into your mind even while you're talking, and you just are like, oh yeah, I want to tell you this. So certainly fun, man. The Mushroom Festival, Oklahoma City. Go check it out online at OK Fungi Festival or OK Mushroom Festival. I think those are the ones. And next week, I've got my class, the OK Calyx Growcast Autumn Immersive, where I'm taking about 30 people out into the woods and I'm showing them what to gather, where to gather, processing it down, and making it and letting them take it home. Y'all, I've got the Candy Natural Farming just finished drying all my bokashi. I've got 2 trash cans full of bokashi. They're going to take every bit of it home. I've got probably 4 totes of IMO 3. They're going to take it home. IMO 4, IMO 5, IMO 1. labs. God, what else? Let's see. Horsetail fermented plant extract. The list goes on. So after this festival, I'm going home and continuing to bottle up 30 bottles of every single thing, 30 bags of everything. I'm going to throw in a ton of genetics. Growcast is going to throw in his stuff, that all the gifts that he can do. So it'll be good. I only think we have tickets left, guys. I think it's sold out, but I'll definitely be posting some videos and some results of how it all went, and it'll be exciting. Also, go to okcalyxshop.com. I've got genetics bundles on sale. I've got all my new cherry stuff, some of the old blueberry stuff, and some of my old cushy stuff, all combined. I've got 3 separate genetics bundles out there, all at different pricing. So if you want a lot of genetics for cheap, these are good. A lot of them are fruity because that's what I like. These are the bundles for you. okcalyxshop.com. All right, Brian.
All right, boys and girls. Until next week, I hope you guys all take the information you've gathered here tonight and apply it. Get those gardens growing. Have a good week, everyone. Good night.